study 9th Standard ICSE Biology Chapter 6 Seeds. Seeds, Structure and Germination. Now, in the previous chapter, we studied how, after fertilization, the ovary becomes the fruit and the ovule becomes the seed. Of course, an ovary may have multiple ovules and hence the fruit will have multiple seeds as well. Now, we shall study about the seed in detail because fertilization is just the start of the journey of a new life. But ultimately, the seed has to germinate in soil to give rise to a new baby plant. So what is a fruit? It's an enlarged and ripened ovary after fertilization. Yes, fruits can grow without fertilization as well. That's called parthenocarpy, but that's very rare. The fruits have a fleshy wall called the pericarp, and it has a seed inside. Inside the seed we have an embryo, and that embryo is a living thing which will grow into a new plant under the right circumstances. The fruit protects the seed and also helps in seed dispersal because animals may eat the entire fruit, and after they travel for a while they may poop out the seed, that is egested, because they can't digest the seeds. Or while eating the fruit the seed might just fall down or may get stuck to the hair of the animals or birds or insects and help to disperse it. Dispersal is very important because if the seeds are going to fall just under its parent tree, then there will be overcrowding in that area and there will be limited resources for all these plants. So hence dispersal is very important. Some seeds can even be dispersed by wind. So what is a seed? It is a ripened ovule. The seed has a seed coat called the testa and the tegmen. If you remember, the ovule before fertilization had coverings called integuments, and now when the ovule has become a seed, the integuments are called the seed coat, that is the testa and the tegmen. They protect the inner parts of the seed, especially the embryo. Now what is a grain? A grain, surprisingly, is a fruit. Here in this fruit I see that there is some fleshy part which has nutrition which we can eat, but the seed coat and the fruit wall are separate. What if the seed coat and the fruit wall are fused together with no fleshy part in between? In such a case, we will call the fruit as a grain, because this grain is not just the fruit but also the seed. It's both. It's a fruit as well as a seed, because the seed and the fruit wall are fused together. There is no fleshy part between the seed coat and the fruit wall. So now we know what is a grain. Example, maize, wheat. Surprisingly, these are all fruits, which is the same as a seed. Now, seeds are of two types, monocotyledonous and dicotyledonous. If you look at a maize grain, you would realize that it has only one cotyledon inside, so it's called monocotyledonous. Even wheat and rice are monocots. Whereas a pea seed, if you've noticed, clearly has a border along which we can split it into two halves. So it has two cotyledons, hence it is called dicotyledonous. Also, seeds may be albuminous or exalbuminous. Albuminous simply means it has endosperm, and exalbuminous means it does not have endosperm. If you remember, during the triple fusion, during fertilization, when the second male gamete fused with the polar nuclei, they became the endosperm, and the female gamete became the embryo. The endosperm serves as a nutritive tissue for the embryo. So, in a way, its function is similar to that of cotyledons. Nutrition, storage of nutrition. But as the seed matures, it may or may not have endosperm left over. If the endosperm still exists, in that case, the cotyledons will be thin. So that's albuminous. But if the endosperm has disappeared, then the cotyledons will become thick, because now they will store a lot of food. So they are called exalbuminous. We shall study their examples soon. Now let's study the diagram of a bean seed in detail. The delta diagram is double ticked, so practice it. You can see that there's testa, the outermost seed coat. There is a tiny hole here called the micropyle. It's a pore which was present even before fertilization. If you notice, the ovule also had this entry point for the pollen tube. The gate through which the two male gametes entered the ovule, that was called micropyle. This micropyle exists even now, and it has a very important function. This micropyle serves as the entry and exit point for respiratory gases and also for water, which is necessary for the germination of the seed. You see the seed coat, the rest of the seed coat is impermeable So the only point of exchange of gases and water is this micropyle for the seed. And this scar that you see, which here is shown as white scar, is called the hilum. This is the point of attachment of the ovule to the placenta in the ovary. So here in this diagram, you can see that there are many ovules attached. I can see 6 ovules attached to the placenta in the ovary. This point of attachment will now be called hilum because the scar remains. In a way, I can say that it is similar to the umbilical cord in human beings. That because umbilical cord was our point of attachment to the placenta of our mother's womb. So that scar which remains behind, if you can call it a scar, in us it is called the belly button. So hilum is like the belly button of a seed. Okay, not the umbilical cord, but the belly button of the seed, the point of attachment. Now the function of testa and tegmen is obviously to protect the seed from injury and attack of bacteria, fungi and insects. Hilum is just a scar left behind. Micropyle's function I've already explained. Now inside the seed, I see a few more parts. I see these cotyledons which store food for the embryo to grow. The embryo itself has a radicle and the plumule. When the embryo starts growing, the radicle will become the root system and the plumule will become the shoot system, that is the stem and the leaves. Now the region of this embryo between this point of attachment of the embryo with the seed and the radicle, so the region between the radicle and this point of attachment, this region is called hypocotyle. And the region between the tip, which is the plumule, and this point of attachment, this region is called epicotyle. So remember these two terms. We'll study this in detail soon. Next, let's talk about the maize grain. So double tick both these diagrams, both have to be studied. External features, well, these are the remains of the style. If you remember, the style is a part of the gynoecium. Endosperm, here's a radicle, here's a plumule. These are actually internal structures. Think of it like an X-ray of a seed, so we can see the inner parts. This is the scutellum. The cotyledon inside a maize grain is called scutellum. Now internally, if I cut a slice of this grain and see the internal parts, it looks like this. The outermost layer is a fused pericarp and testa, that is the seed coat and the fruit wall fused together. That is why it's called a grain, because the fruit and the seed is one and the same here. Just inner to it is the aileron layer, which is a protein-rich layer. This is endosperm, which stores food. So clearly this is an albuminous seed because it has endosperm persisting even now. It has a cotyledon as well, but it's very thin, obviously, and it's called scutellum. Now this region is called the embryo. The embryo has a radicle, which will grow into the root in the future, and it has a plumule, which will grow into the shoot in the future. In between, there is an epithelium layer to separate the embryo and the cotyledon from the endosperm. Now there is a thin layer of protective sheet on top of plumule as well as on top of the radicle. They are called coleoptile and coleorhiza respectively. So together this is the embryo, which will grow into the baby plant called the maize plant when germination occurs. So remember, maize grain is a monocot and it's endospermic. This is true even for rice, wheat and oat. Now let us understand the difference between bean and maize. Bean is dicot, maize is one cot, cotyledon, that is monocot, called the scutellum. Bean is exalbuminous, it does not have an endosperm, it has large cotyledons, that's why. Maize, on the other hand, is albuminous, it has a large endosperm and a very thin cotyledon. In bean, the embryo is quite large, as you can see here. And in maize, the embryo is quite small, comparatively smaller. Here, the plumule's leaves are folded, whereas the plumule leaves are rolled. The plumule is large and small. Hilum and micropyle are visible. You could see the hilum and micropyle in the seed. But in a maize grain, you can't clearly see the hilum and micropyle. And finally, in the bean, the seed is separately contained in the fruit, which is called the pod. Yes, the pod in which the beans exist is a fruit. Even for peas, for example, the green peas, the green pod is actually the fruit. Yes, it does not have any fleshy part inside, and sometimes it may even have worms inside, but that is a fruit, and the individual peas are seeds, and those seeds have in them life, a baby, a future baby pea plant, but we eat it. Maize, it's a single grain because it's a fusion of the fruit and the seed both. So when the seeds get appropriate conditions, they start germinating. Let's study about germination. Now, when a seed is formed, immediately if we take the seed and put it in the soil, it will not germinate because it is in a dormant state. You see, the seed has to first mature. The embryo has to mature. Then only germination can happen. So this phase is called a dormancy phase, which may last for some time depending on the species. Yes, we can chemically wake them up from dormancy and force them to start growing. Otherwise, they will remain dormant till they are mature. But even if they are dormant, do not think that they are non-living, because the embryo is living. Little bit of metabolism is happening inside. They are using the food stored inside the seed to survive, to use the oxygen, to give out the carbon dioxide and produce some heat as well. We can store seeds in a dormant state for decades, even a century. So there are certain seeds, if frozen, totally frozen for 100 years, and then if we sow them in the soil, they will still have the ability to grow into a new plant. It's as if they are hibernating. So what is germination? Germination is a process of formation of a seedling from the embryo. That is, the embryo should grow and the radicals and the plumules should form a very small plant. That is called a seedling. It's called a seedling because the tiny plant is still dependent on the seed for food. It doesn't yet have its own leaves to do photosynthesis. Once the plant has its own leaves, the seed would have withered away. All the food would have been used up. The cotyledons, the endosperm all disappear. And now we have a baby plant which is independent. It cannot make its own food, and that is called a plantlet. So now we know the difference between a seedling and a plantlet. But what are the conditions necessary for germination? It's common sense that water is very important for germination, because water is required in two ways for the seed. The water is absorbed by the micropyle as well as by the seed coat through imbibition. That's a process of just attracting water, which we study in 10th standard. That helps the seed coat to rupture. Only if the seed coat breaks apart can the radical and the plumule come out. And the second reason is water is a solvent for many chemicals, for food, for enzymes. So if these chemicals have to be transported inside the seed, water provides the necessary medium for the diffusion of these substances. Next condition is suitable temperature. The optimum temperature is 25 to 35 degrees Celsius. You see, above 35 degrees Celsius, the enzymes start getting destroyed. In fact, above 40 degrees Celsius, enzymes cannot function anymore. And without enzymes, life cannot exist. So this is the optimum temperature. However, the tropical plants, they grow best at a higher temperature rather than the lower temperature. So it depends from plant to plant. In very cold temperatures, the seeds may not germinate at all. That's why if you keep the seeds in the refrigerator, they will stay like that for a very long time. Next, they need oxygen. Obviously, oxygen is important for respiration, to give them enough energy to grow. So when seeds are grown very deep into the soil, they might not germinate at all for two reasons. Because there is, first of all, no proper supply of oxygen for respiration. And secondly, if they're very deep inside the soil, then they cannot produce so much force to break through the soil and come out to the earth. There is insufficient pushing force in the embryonic parts to break through the upper layers of soil. It's like this. If you are covered in some sand, you can easily come out of it. But if you are buried 6 feet under, it'll be very difficult for you to come out, because then a lot of force will be required. Now, some experiments with germination. The first experiment is extremely complicated, so I need your full attention here. What is the proof that water is necessary for germination? Now, it's a tricky one. How can we prove that water is necessary for germination? What we'll do is we'll take 2 beakers. There's no diagram available as well, so you'll have to imagine them. On a wet cotton wool, put some seeds, and on a dry cotton wool, put some other seeds. You will realize that the wet cotton wool seeds will germinate first. In fact, the dry cotton wool may not germinate at all. Those seeds will not germinate. Thus, it proves that water is necessary for germination. Next experiment. How to prove that a suitable temperature is necessary for germination? Now, this is a classic, tough one. You can keep some seeds in a beaker A and put them in the ordinary room temperature, and in beaker B, the seeds are kept in the refrigerator. Guess which one will germinate? The ordinary temperature one, hence proved. Now, the third experiment. Students, you need to read all these experiments. You need to read the whole textbook. I'm just explaining the concepts here. Now, experiment to prove that oxygen is necessary for germination. So I have wet cotton wool on both of them. So both of them are getting moisture. Here I keep plain water. That's just a control. And this is the actual experimental setup where we have alkaline pyrogallic acid. Alkaline acid, that's a bit of a contradiction, isn't it? That's what happens when biology has chemistry involved in it. Now, alkaline pyrogallic acid has a property that it can absorb all the oxygen inside this flask. Once all the oxygen is absorbed, and of course no oxygen can enter from outside, then the seeds will not germinate, whereas these will because they have oxygen out here in the air. This proves that oxygen is required for germination. However, you will notice that after a long time, after many days, even these seeds will start germinating a bit. That's because seeds have the capacity to do anaerobic respiration. They can do respiration even without oxygen. And in such a case, they don't produce the normal water molecules, rather they produce ethyl alcohol. More about anaerobic respiration in a different chapter. That is the next chapter, 7. And now the 3 bean seeds experiment. In this experiment, I see that the bottom seed is getting plenty of water, but it is not exposed to air. The middle seed is getting the best of both the worlds, and the top seed is getting air, but not plenty of water. We will notice that the middle seed germinates fastest. The bottom seed will germinate very slowly because there is some oxygen dissolved in water. And the top seed will also germinate very slowly because there is some moisture available in air. But the middle seed germinates fastest, clearly showing that water and oxygen are both required for germination, although the requirement of oxygen is not fully demonstrated in this experiment. Next, let's talk about the types of germination. Now, if you look at these 2 diagrams, here we have a pea seed, and here we have a bean seed. Let's go step by step. The radicle is growing. Now I see that this is the radicle and this is the plumule, okay? And the region in between here, it's called epicotyle, and this is called hypocotyle, if you remember. I had talked about it in this diagram. This region till the radicle is called hypocotyle, and this region till the plumule is called epicotyle. So here I see that the epicotyle elongates first. The epicotyle and hypocotyle both do not elongate at the same time. No, one of them elongates first. In the case of P, the epicotyle elongates first, pushing the plumule out of the ground. So the cotyledon remains under the surface of the earth. And of course later on the hypocotyle also grows. But first the epicotyle grew. Such germination is called hypogeal germination. Hypo means under. So since the cotyledon remains underground, or maybe just on the surface, it is called hypogeal germination. Now compare this with bean seed. Here I see that the hypocotyle is elongating first. The hypocotyle is elongating first, so the cotyledon is pushed out of the soil. And then later the epicotyle starts growing. So here the cotyledons are above the surface, hence this type of germination is called epigeal germination. So the difference between the two is that in hypogeal, cotyledons are underground. Remember, hypo means under. And in epigeal germination, the cotyledons are pushed above the ground because epi means above. Also remember, in hypogeal germination, epicotyle elongates first, and in epigeal germination, hypocotyle elongates first. And remember the examples, P and B. Now look at this diagram. Only look at the diagram, don't read the text, and tell me by looking at the diagram without reading the text, tell me, is this hypogeal or epigeal germination? Well, I see the biggest hint is that the cotyledon is still underground. That means it's hypogeal. And also I see that the epicotyle is elongating first. The coleoptile has been pushed up. If you remember, coleoptile was the layer, the tissue, the sheath protecting the plumule. It was there in the maize grain diagram, coleoptile. Here we have the definitions of epicotyle and hypocotyle. Next, viviparous germination is not in syllabus, but just for your information, it is a germination when the seed or the fruit is still attached to the plant. Usually, the fruits fall off and then they start germinating, the seeds start germinating. But sometimes, even though the fruit is attached to the plant, it may start germinating. This reminds me of kangaroos. The babies are still stuck to the the mother, and in the pouch they continue to grow. Next, the seedling. I've already explained it. Seedling is a stage in the growth of a plant from a seed before it has become wholly independent of the food stored in it. And once it has used up all the food of the seed and it has grown its own leaves and does photosynthesis, then it is called a plantlet. Hi students, this is AJ sir. If you like this video, press the like button. If you would like to enroll for my online test series or online lectures, email me or message me on Instagram. Check the description for more information.